without further ado, thinking anthropologically about astrobiology. So, how do you define astrobiology? This is one of the most important questions I ask during my interviews while conducting my fieldwork. And while it may seem very general, when scientists would start to answer this question, it kind of spurred conversations about, maybe a moment they had in their graduate student training where they had to come up with the answer to this question, or a recent experiment in which they had to think about the question of astrobiology a little bit differently. And so this question also gets at these multiple scales of what doing astrobiology is, as a science, as an institution, and as an individual practice. So I started my research back in a long time ago, but 2018 was the first kind of like moment when I started thinking about these astrobiological questions at a planetary protection training session. And then I moved on into the archives at NASA HQ in 2019. And I'm sure you won't find it really difficult to imagine that their definitions of astrobiology are vastly different, related. But what they what they kind of contextualize astrobiology as, and how the implications for doing astrobiological work was different. And as I'll explain a little bit later, these differences across different institutions, as I've been doing my research, NASA Ames to the archives, have different reverberations of how the practice of astrobiology is actually done. So if, because every talk should have a participatory element, if you feel like sharing or feel like participating, I encourage you to write in the chat how you define astrobiology based on these kind of three questions, either examples from your work, how you do your work, or what methods you use, or how you inform yourself about astrobiology. And I promise I will remember to come back to it at the end of the presentation. So I am speaking from the anthropology of outer space. And this is a field that has been gaining more attention over the last 20 years. Anthropology has had many manifestations, and particularly over the decades, it's become more attuned to the practices and processes that people engage in, as well as these like living experiences of institutions, or are interconnected with interconnections and interactions with non-humans or the environment or technology. And in the case of my work, the interplanetary, in defining these, and it has implications for defining important aspects of our human or earthly experiences. So I'm going to take a while, and so bear with me here to kind of explain the abstract theoretical foundation of my work before diving into the more exciting examples. And so for the purpose of this talk, the facet of my research question that I want to focus on is the one you see right here. What are the effects or implications of the doing of astrobiology on the unfolding of astrobiology as a scientific discipline? And for a bit of background, really quickly, in anthropology, in order to complete your degree, you have 3 years of coursework followed by a comprehensive exam and a thesis proposal. And then we're let loose into what we call the field, which is the locations where our central research question may be addressed. Usually take about 1 to 2 years to, be in the field, gather as much data as we can before we go back to our universities to write our dissertations. And so over the course of my research, this central question that you see has gone through multiple permutations as the ideas that I had initially in coursework shifted as I met scientists, participated in workshops and conferences, and had the experience to actually know what astrobiology was in the doing. And so I'm going to take you through this journey with me as like these different experiences have shaped what I've come to think about astrobiology. So as I mentioned, I did 8 months of fieldwork at the NASA HQ archives in the Library of Congress. And there I was trying to trace like these early historical efforts of exobiology. And it was in finding these like slightly snarky correspondence between scientists at different NASA facilities, along with these polite arguments or assertions about what the best decontamination methods were, or these like massive organizational charts of who responded to who and who was in charge of what department, as well as many, many, many technical reports that I wanted to start to focus on this question of epistemologies. the accepted knowledge about what astrobiology was and how it did it, and to poke at how astrobiology was configured, how these configurations and reconfigurations happened across time and space, and what the experience of it was. And in recognizing that there's a relationship between questions and the sites I had access to and the people I collaborated with, I also ask about these implications of how these relational trends are expressed. And in so doing, I ask several different questions, such as, what are the methodological strategies that underpin astrobiology and allow for astrobiology to be considered science? What questions about, for example, what questions about the search of life are sensible to ask? Or what factors or constraints are expected to be considered? What missions become acceptable? What perspectives are deemed less popular? And then something else I'm interested in, which is, how does this knowledge about astrobiology become expressed and circulated? Who has authority over this circulation and who has access to it? On the other hand, as I'll go into, I also attend to what I call the lived experience of science. The iterations when you're going over a hypothesis in a proposal, the different times it changes in order to get closer and closer to what you want it to be, or in a discussion of, let's say, biosignatures, the topics that reach really quick consensus versus the ones that have a lot of debate, and which experiments become more validated than the other. These instances of pause, creativity, speculation, assertion, and doubt are just as significant in the progress, practice, and process of science as the reports or decadal survey, white papers, or meeting minutes. So let's move on to the field. I had the wonderful privilege of going to the Atacama Desert with a group of scientists from NASA Ames and Johns Hopkins University. And the purpose of this trip was to download data from multiple sensors that they had planted previously in halites or salt rock that were known to host microbial communities powered by these photosynthetic microbes. And the data from within these sensors would be compared with previous trips from the Atacama, as well as analyses run on environmental data, such as the air, wet-dry cycle patterns, light duration, so forth. In addition, these scientists wanted to gather samples of these microbial communities in order to run RNA analyses and to look at the different pathways that could be tied to microbial community, such as membrane repair. So I understood these halites and their significance to be twofold, right? They were one of the last remaining locations and environments in which life eked out an existence in the driest hot desert on Earth, and that studying these communities could tell us about the relationship between three core life requirements: energy, nutrients, and water. And two, if understanding the relationship between environmental conditions, their extremity, and how limited microbial life was could be discovered, then this could perhaps help ask questions about other environments, such as Mars, or, other places on Earth where life may have utilized similar strategies or thing like that. And a part of getting to this data was through sampling. And so one of my favorite moments in the field was learning how to sample properly. So we had the switchblade and we had to scrape at the halites and the inside in order to remove some of the material. And I learned how to twist a knife in order to get more material. I learned how to use my body as like a wind shield, because at 1 o'clock every day the wind would pick up, and if you weren't careful, your sample would spill out all over the place. And I also learned, how to find this greenish hue to tell where the microbes were kind of hiding out, as I like to think about it, and how to approximate what 30 grams of sample was. And this was part of the tactile experiential knowledge that I picked up while doing astrobiology at the site. While scientists were gathering data to address questions about activity of microbial communities, I was collecting data about these methods they were employing. And I often did it through these different types of conversations, publications, explanations of scientific data or how to sample, as well as these experiential textual moments such as sampling, finding halite sample sites because the sensors were very difficult to find, or reading these scribbled maps in the back of a car as we're driving along these endless roads. And this was all part of my of gaining this understanding of what actually goes on in the Atacama. So lingering on this question of methodological strategies, the data from the field, from the Atacama, as well as what I picked up in the archives, conferences, my time at NASA Ames, workshops such as PC3 or the knowledge base, have begun to fall into two different categories. There's crafting definitions and setting of detection thresholds. And definition making, like how do you define a biosignature or a habitable environment, and then detection limits such as, what measurements do you need in order to get a proper reading, or what is the signal to noise ratio. And these various methodological strategies were utilized in order to craft these definitions or detection thresholds. And doing so touched on these following questions. How should a definition of X be made? How is it being defined, and what are the boundaries of those decisions? In the desert, for example, there's the question of what balance of water, energy, and nutrients allowed microbes to survive, and how did you define that balance? Oftentimes, fieldwork would also, in making these definitions, you'd have to rely on certain publications or experts to invoke, to justify or give weight to how a definition or threshold should be set, and so forth. In observing these activities, another set of data also arose about the implications of these definitions or detection thresholds, exemplified by these questions. These implications included what instruments should be selected that fit most effectively with the particular scientific goal, or the measurements that had to be taken that would be effective for answering these questions. Or thinking about the instrument also required thinking about how it would be constructed, by whom, and with what funds. So again, thinking with the Atacama, the placement of the sensors and the ability to sample enough of the halite for RNA analysis are themselves derived from efforts to define the parameters of microbial survival. And these have implications for where the research with the halite will go next. Thus, the movement between the definition being made and the ways in which it's defined, and then the implications of both its operationalization as well as the shifts that may happen as it's occurring, in terms of, let's say, suddenly you now need to think about sample availability or the particular sensitivity of an instrument, all reverberate in the space of conducting astrobiology. What these patterns and feedback loops of activity lead me to ask is this final question. What do definitions and detections tell me about science in action and narratives about science? And what is this relationship between the two? And of course, this movement between answering or addressing this final question also reverberates back into this feed loop of, what are the methodological strategies of defining or detecting? Let's return again to the desert. One of the most popular topics of conversation was, how did these microbial communities, survive? There was an extreme limit of water, tons of sunlight, and unknown quantity or access to nutrients, so how did they do it? And at the time, I wasn't particularly well versed in previous research done in the Atacama. I'd done as much reading as I could, but I didn't really understand it. And one of the most powerful ways that I gained this understanding are through these following stories. So during a conversation, the microbes in the halite were described to me by one scientist as living in an ocean-like environment. And I'd never really thought about microbes in such a way. And I know it seems like a really simple story at the end, ocean environment, but that had the effect of kind of shaking me and reorienting my perspective so I can think about the microbes in the halite on a different scale. Also, during one of our lunch breaks, we had this speculative conversation in which someone asked if we could build any instrument that you can make that was not restricted by the laws of physics, what would it be? And one of the scientists said that they would make a machine that could shrink you to the size of a microbe and you could see what's happening at their level. Of course, with the proper protections of so you don't get caught up in, nutrient wars. And these imaginative conversations were just as effective, at least for me, to decenter this human perspective that I had about microbes and reposition myself into a frame of mind that could better translate this microbial existence into something I could relate to. And then I was able to take a step back and begin to ask, okay, so what about the microbial environmental relationship should I pay attention to? How important are these interactions for kind of understanding these larger questions of survival in the desert? So for me, to put together scientific sensor data, the theoretical debates about microbial survival, and the speculative musings about what actually goes down in the microbial community, and to begin to understand anthropologically meant holding not only these events and processes in the field as data, but also keeping in mind, as I spoke about before, these relationships and implications of the data to each other and to the scientists producing it, and then also to astrobiology at large. So what does this mean? It means something called, in anthropology, building the assemblage. And for me, I define an assemblage as the interconnections between people, events, objects, interactions, processes that congeal or coalesce around anything, in this case, the Atacama. And building an assemblage of what constitutes astrobiology in the Atacama, I held all these tensions and stories and questions together. So I had in this assemblage thinking from the perspective of a microbe. I also had, my experiences in the field of driving to the different sites, of sampling, of looking for these sensors. The assemblage also includes movements or patterns that emerge from efforts to set definition and detection limits, as I've spoken about, as well as the people who were involved in producing this knowledge about the halides. The utility of the assemblage, rather than, say, listing a list of factors or drawing lines between one person and one object, is that the assemblage is dynamic. Lines from one factor to another may change or affect each other differently. The assemblage asks us to consider the context that these actions are taking place. So, for example, the context of the Atacama, and how the context itself is shaped not only by us, or the scientists who are taking samples in the field, but also other researchers who may be there, or the mining activities, or any number of people who are passing through that space. And so the power of thinking of the assemblage is that you can take these relations betwixt and between and kind of put it into the context of what I described as this movement of crafting definitions and for these larger questions about astrobiology. Which leads me to my next fieldwork example, which was a phenomenal experience of Orbilander. So from January through July 2020, I had a really wonderful opportunity to work with a group of scientists from APL, Ames, Goddard, and JPL on what became the Enceladus Orbilander flagship mission concept study for the Decadal Survey. And Orbilander was one of 4 spacecraft architectures that were constructed during this time to do science at Enceladus, and Orbilander was selected as a mix of an orbiter and a lander because it was deemed most effective and for achieving the science goals. And this image you see right here is the fact sheet of the completed report. And I really can't emphasize this enough. Each title that you see, each bold text, each bullet point, and the 2 images of the orbiter and of the lander you see here were the culmination of hours and weeks and months of discussion and email exchanges and individuals taking the lead on a certain characteristic and doing a deep dive and reporting back to the group. And I'm sure you can empathize if you've ever written a report or are coming up with a grant proposal, having to minimize or simplify an immense amount of data into a bullet point is, quite an effort. And what I want to highlight in this is these science objectives. So I can't tell you the number of times I heard the phrase during these couple months, let's go back to the science. Or what does this measurement or have to do or becomes affected by battery constraint or mass constraint? And how does that address the science question? And these kind of constant referrals back to the science objective, science question, science goal, flagged for me not only the underlying principle of the researchers I was working with, but also, you know, alerted me to the different practices that allowed for those science objectives to become prominent or allowed them to happen in the course of constructing this missions concept study. And a lot of the other aspects of, let's say, power or battery constraint or data, having enough data to send back to Earth were negotiations that happen in the trade space or the space of the mission. And all this to say, and I'm vastly oversimplifying this experience and how exciting it was to kind of think through these questions, was that with working with the Orbilander science team and then observing how that science became translated into an actual payload and spacecraft design with the engineers, was a chance for me to see another assemblage come into form, that around Orbilander and the relations that happened between those nodes at APL or Ames or Goddard or JPL. And it's the work of my dissertation to bring the assemblage around Orbilander and the one around Atacama Desert together. Which brings me to this lovely and complex chart, which is, I know it's hard to read, but it is the heart of what I'm building towards as one of my central theories. And so from the perspective of my fieldwork, what I'm calling gravity wells is essentially an assemblage in space-time, because the space metaphor is too good to pass up. And it's this idea of that everything I've observed in the field, from the archives, the Atacama, to different online or in-person conferences back in the day, as well as the Planetary Protection Workshop, the decadal survey reviews, my sit-ins on the exobiology branch meetings at Ames, the PCE3 workshops, CLD town halls or SMD town halls. All of these have a different mass and a different pull and are connected to each other in different ways. The space-time element recognizes that all that I've observed occurred across different spaces and different times. And so, for instance, something like defining a potential measurement to take on Mars has occurred repeatedly throughout time and space for Viking in the 1970s and is happening right now with Perseverance. And I see gravity wells as a productive way of seeing how the many threads to astrobiology and its configuration as a science come together and become coherent-ish, and how that seeming coherence in the moment is made up of a lot of moving parts that are not necessarily temporally or spatially bound. So take the very simple example of defining a biosignature. It has, as this slide is showing, it has multiple different kind of pulls between previous publications to fieldwork and analog sites like the Atacama, to nominal and planned missions like Orbilander, as well as sampling requirements, planetary protection protocols, different other contamination issues, ongoing or missions, technological innovations in both the commercial sector as well as at NASA, and all of these kind of pull and morph and shape and orbit around each other. And is something that I think is important to kind of grapple with in order to see this larger picture of astrobiology. And you're in there too, now that I've done this talk, or I'm doing this talk, and of course I am there. Which gets me to more of like the anthropological perspective that I always bring with me, which is that part of the gravity well, it can't exist separately from the people who are enacting these practices or these missions or what have you. To be situated, what I refer to here as situatedness, is something that scholars like Donna Haraway recognize is as the lived particular context that we are in, that we embody, that we experience. And our perspective comes from our position. In this case, for me, I'm an anthropologist. I'm a grad student. I'm a woman of color. More often than not, I'm pretty Zoom fatigued, and I'm picking up astrobiology and biology as I go because I don't have formal training in the science, but I've taken a couple classes and I've learned a lot from being in the field. And I can never have an experience or a perspective that is encompassing of all human or non-human experience or their identity. And while my experience may expand as I encounter different people or different contexts, I can never know everything. So being situated means also always having a partial perspective. And it's with this partiality that we experience and navigate the world. Which brings me back to the chat, which I'm not actually sure I'm going to be able to see. So maybe I'll hold this conversation for something later, but I imagine that, you know, entries were, in defining astrobiology may have been similar in some cases, maybe defining it as a search for evidence of life or in the context of your lab or a particular project. And what I intended to highlight with this was that while we may have similar training or be in the same project in defining astrobiology, and maybe those definitions look really awfully similar, the experience of it is different and distinct for each of you. And I think that over time, those differences are what those small shifts in astrobiology over time are indicative of. And I'll talk more about that very shortly. I do want to mention what I call here multi-sited. My research is multi-sited. It took place in a lot of different places and kind of decentralizes something that anthropology is kind of founded on, which is you do work in one place of one people. And I don't necessarily see that as something that is true. So being multi-sited means that I can decentralize the location from which astrobiology is quote unquote defined or determined. There are flows and frictions across these different hubs. And you can see that by how different NASA facilities or different universities may talk with each other about different issues or different questions, or how astrobiology may be conducted in an academic setting versus a NASA setting versus commercially. And each site offers a different perspective. So notice these images. The first is a picture of the NASA Ames Visitors Center tent, and it was the closest I could get to Ames in April of 2020 when the center closed with the pandemic. The second image is of the Salar Grande in the Atacama, and literally what you see are miles and miles and miles of halides stretching into the distance. It was unreal, and this was in March of 2020. The NASA archives in the main HQ building is the third image, and located in the basement, if you don't know, two rights from the elevator. And it is a really wonderful, interesting place. And I believe the folder I was looking at was probably from the 1960s or 1970s. And then the final image is of a conference in the DC Convention Center. And to be honest, I don't remember what conference it was. I do know that it was in DC because of the carpet. It's kind of distinct and you don't forget it. And that was probably in January of 2019 when I did a lot of my conference visits. And all this to say is that, from each of these locations, I gained a different perspective of astrobiology, and each of these had their own assemblages that now are kind of floating around or situated in this gravity well that I call my research. And finally, theory from the ground, and I'll only briefly touch on this, is my kind of theoretical claim that I am making as I begin to write. And it's essentially that since I speak from my own perspective and I've only had access to certain locations from which I've gained their perspective on astrobiology, why would I turn around and then go to a French theorist or anthropologist to use their theory from the 1970s to analyze my data? And while I won't completely cut off all theory from the past, because there is a lot of useful things that I've learned from anthropologists or science and technology scholars about how to do an anthropology of science, I am going to put it together with a lot of the theories and patterns I've observed from conversations or experiences in order to create this balance of theory from the ground and theory from previous scholarship. So stay tuned. It's something I'm thinking a lot about right now. So I did want to touch on lessons in learning because one of my kind of stakes in the ground as a scholar is discussing methodology and kind of making sure that we have these discussions of what it's like to do our work and instead of just hiding it behind the results of what we're working on. And I learned a lot in the field. And I was speaking to Erin about this before the talk, but my phone is filled with, because I got tired of writing, recordings of myself talking about these moments when, suddenly something in the field makes sense and I'm out walking and I have to make sure that I don't forget it. Or, ruminations and reflections on what I could have done differently in an interview or what I should go to next as the next seminar or Zoom conference. And so one of the lessons I've learned a lot is in making these field notes, these compilations of notes about the field. And usually these notes help me to contextualize events or interviews or observations by taking note of the shapes and colors of a room or the patterns in a carpet, right? The expressions on people's faces, whether people are secretly on their phone during a conference or falling asleep. But in the Zoom world, that has offered some really interesting questions because I'm now looking into someone's living room, which was not at all part of the research questions that I was hoping to answer. And it's made me think a lot about what is how far does the field reach? What are my responsibilities as a researcher? Um, and how do I kind of balance the demands of what I'm expected to deliver as a dissertation with, the conversations I have with scientists who I, you know, really admire their work or think what they're asking is really interesting, and I don't want to have any sort of broach in privacy because, you know, Zoom World offers a mirror into everyone's background and personal life. So this is something that digital ethnographers think a lot about, and I'm sure other people do, but it's something that I thought a lot for how methodology I should write about this in my work. And it's also made me think a lot about in-person interactions. I started my work in California at Ames in March, and so a lot of the people I've met and spent time with over meetings over the last several months, I probably won't meet in person, or at least not anytime soon. And that's a weird realization to have when everything that I've been taught about anthropology is that you are in the same place as a person. You have conversations with these people and you get to know them. So this has also been something I've been grappling with as, I still was able to conduct my fieldwork. What are the implications for how anthropology should be shifting and changing to accommodate, different points of access and the world, the state of the world? And then finally, kind of going back to this idea, this theory of partial perspectives is as it pertains to my own work. So when I was beginning research, I had this constant worry that I wasn't talking to enough people, that in order to justify what I was doing to my grants, I had to make sure that I spoke to everyone and possible, went to every single Zoom meeting. And, having a partial perspective made me realize that I can't talk to everyone, can't be everywhere. And I think that's a really powerful thing because it re-centers scholarship around not this abstract perspective of like what can this question answer, but what can this question answer from the perspective and situated positions that we occupy, which I think is a lot more realistic. So finally, my preliminary conclusions, and they're very preliminary and very partial. The um, as I was kind of putting this talk together, I began to see that astrobiology as a science can be splintered into at least two different kind of assemblages, right? There's this science as an authoritative or coherent institution that is configured by reports and bureaucracy and sample requirements, which in themselves are lived experiences and messy and come together at the last minute, but in going towards the narrative or institution of science seem to gain this this coherent picture or this coalesced congealed picture that then become a reference point for others who are coming up with missions or experiments. And then, as I've discussed before, there's this lived aspect of science in action and the activities and conversations and debates that happen around the Zoom table as scientists try to define a biosignature or how best to go about detecting, this particular measurement based on considerations or constraints. And this is what I've kind of tried to visualize in the gravity well, that science is mutable and partial and is only um, only emerges through interactions. And I think there's this really interesting space in between that as scientists are going about defining or detecting, they have to mold it into a publication or a proposal for a mission in order for it to be funded or selected or approved. And this is kind of expressed with this final question. What is the space between this coherent or coherenting, I should probably say, narrative of science and the gravity well of scientific activity? And going forward in my analysis, I will see if and what my data tells me about the shape of the space. But I think for now, what I want to kind of leave or end this talk with is like highlighting this tension between narratives and institutions of science and histories of science with this gravity well experience, and how productions in shifts in what becomes sensible astrobiological questions, what becomes validated, or what something becomes that we aspire towards are kind of produced in this in-between space, as you tag between the narrative and the coherent with the mutable experiential. And this in-between space, I believe, is a way to start understanding, the patterns that we see in the past of astrobiology, at least partially because we don't know all the factors and we never can, such as like what people were thinking at the moment. But also maybe we can start to understand the patterns going forward as people like you are doing astrobiology for a future. What questions are you bringing to your work? What perspectives and new methods and ways of writing proposals may be ever so minutely shifting the field so that the astrobiology that you are experiencing today may be slightly different or ask for certain different partners, maybe like anthropologists, in the future. And I think that's where I'm going to end it. So thank you everyone for listening. Thank you, Georgia Tech and Aaron and Dr. Schmidt for inviting me, as well as the scientists I did my work with in Chile, the Orbilander mission, a lot of feedback I got with Enfold, and then my funders. So any